Hello and welcome to this session on the genetic abnormalities in hematologic malignancies. I am Jeevan, and for the last decade or so I have been living on this tiny island of Nevis. So I am going to be discussing the genetics of hematologic malignancies. We will focus mainly on the genetic abnormalities in the leukemias and lymphomas. Uh, we will try to, hopefully by the end of session, become familiar with some of the more common abnormalities that we see, and especially some of the favorable and the unfavorable abnormalities associated with acute leukemias. We will also focus on some very specific mutations, particularly the translocation associated with the acute promyelocytic leukemia. We'll also be discussing the role of the Philadelphia chromosome in chronic myeloid leukemia, the JAK2 mutation in polycythemia vera, and also the several mutations that are seen with myelodysplastic syndromes, or as the new literature suggests, myelodysplastic neoplasms. And lastly, we will try to understand some of the translocations that are associated particularly with non-Hodgkin lymphomas. So, why do we need to understand these mutations in hematologic malignancies? The mutations work in different ways. Uh, Many of these mutations are actually translocations, but as we can see from this image, these mutations can work in different ways. In many cases, the mutations can be pro-growth mutation. So, particularly the MYC, which is a transcription factor. So, if there is a translocation and it is amplified, it could cause more cell division, thereby leading to a proliferating cell population. Sometimes the mutation influence self-renewal. They cause these stem cells to keep self-renewing, and that may also be associated with a maturation arrest. This will also lead to a neoplastic proliferation. There is a third mechanism that may be involved. We see that specifically in the case of follicular lymphoma, where there is no increase in the proliferation of cells, but because these cells stop dying, the neoplastic cells stop dying, they accumulate and result in tumors. So mutations can work in different ways. Either they cause more growth of the cells, or they cause more self-renewal of the stem cells, or they decrease the apoptosis of the tumor cells. And in terms of the malignancies, we will look at some of the more common malignancies. These include uh, tumors arising from the lymphoid lineage or from the myeloid lineage, and there is also the histiocytosis. It is interesting that in case of myeloid neoplasms, all of them would arise from the bone marrow. So we have the acute myeloid leukemia, we have the myeloproliferative neoplasms, including the chronic myeloid leukemia, and then there is the myelodysplastic syndromes. However, in the case of lymphoid neoplasms, They could either arise from the bone marrow, in which case we would label them as leukemias, or they could arise from extramedullary sites, more commonly the lymph nodes, but they could also arise from other lymphoid organs like the spleen or mucosa associated lymphoid tissue. Those proliferations, we would call them as lymphomas. What is also interesting is that mutations work both ways. If you look at these three images, the first image here shows a normal bone marrow. You look at the bone marrow, you see those fat cells. You see a mixed population of cells of the different lineages. You see erythroid cells, myeloid cells, mature cells, immature cells, precursors. And in B, what we see is there seems to be more of one particular kind of cell that is trying to replace the entire marrows. And this is a good example of what happens with acute leukemias. A monotonous population of immature cells start to occupy the entire marrow. The last slide shows a decrease in the cell population. What is interesting is a mutation can either lead to a decrease or an increase. In this case, of course, we are focusing on malignancies. So we will try to understand how mutations lead to acute leukemias and how they are how our understanding of these genetic abnormalities can help us devise better treatment strategies, more effective treatment strategies. As I've already mentioned, even though the population of tumor cells may be similar in the lymphoid leukemia and the lymphoid lymphomas, the criteria that we use to differentiate between a lymphoma and a leukemia is where did the tumor originate. If the tumor originates in the bone marrow, we call it leukemia. This can either be myeloid or lymphoid. It can either be acute or chronic. On the other hand, lymphoma could start in lymph nodes, it could start in the spleen, it could start in other lymphoid collections like gut-associated lymphoid tissue, mucosa-associated lymphoid tissue. But this can only be lymphoid. This, however, is further divided into the Hodgkin or the non-Hodgkin lymphomas. Let's start with the acute leukemias. In acute leukemias, the most common mutations are driver mutations. These mutations are usually gene rearrangements or point substitutions, which basically interfere with those transcription factors that normally regulate the differentiation of the stem cells. A very specific example being the transcription factor PAX5. This transcription factor is required for the differentiation of the early B cell progenitors. So what if there is a mutation that affects this transcription factor and it is not available? The B cell proliferation would stop at the early B cell and result in the most common form of acute lymphoblastic leukemia of the B cell lineage. Besides these driver mutations affecting signaling molecules like tyrosine kinases and the RAS signaling pathway, they will lead to increased proliferation of these cells, also increased survival. And increasingly, we are learning about the epigenetic alterations also, especially those affecting the factors that regulate the histones. The myeloid neoplasms, we generally would approach them in terms of the acute myeloid leukemias, and as you are all aware, the criteria for diagnosis is more than 20% blasts, myeloid blasts in the bone marrow. And then there is the chronic myeloproliferative disorders like chronic myeloid leukemia, polycythemia vera, myelofibrosis. There is an inappropriate increase in the production, but these appear to resemble normal, mature looking cells. And then there is the myelodysplastic syndrome, where there is a clonal proliferation of these stem cells, which leads to ineffective erythropoiesis and therefore refractory cytopenias. Right. Uh, what we see here is a peripheral smear showing a lot of very large looking cells, and especially when we compare with the RBCs in the background. So, since we know that the usual size of an RBC is somewhere between 6 to 8 microns, what we are noticing is this very large cells. They have very large nuclei. Uh, Also, they seem to have a moderate amount of cytoplasm. What is also prominent is the granules. In fact, in one particular cell, we can even see that the granules have condensed to form the so-called OR rod. You also notice that these large nuclei within the cells have prominent looking nucleoli. Basically, these are mostly promyelocytes, and this is a case of acute myeloid leukemia. Now, acute myeloid leukemia has a lot of subdivisions. Uh, it is a little bit, it can be a little bit challenging to understand the different subdivisions, but now that we have the WHO classification, things have become a little more simple. It's a little more easier to approach the acute myeloid leukemias. What we see here is that the four classes of the acute myeloid leukemia is basically, or largely, this is the class of the most interest, the AMLs that are associated with specific genetic aberrations. And why so much emphasis on these specific genetic aberrations? Because of the way they influence prognosis. Right? There are three translocations, as we can see, which are associated with favorable prognosis. And there is the MLL fusion, which is associated with poor prognosis. You also notice most of the ones associated with dysplasia or therapy-related AMLs, they carry very poor prognosis. In general, AMLs have very bad outcomes. Most patients who are diagnosed with AML do not have long-term survivals. And therefore, especially in the case of AML, the favorable translocations have even more importance. We will look at one very specific example of AML with a very particular translocation that has favorable prognosis. There is the fourth class where we still use morphology as the basis of defining the acute leukemias. Although, as I said, the class that is perhaps become most relevant now is the ones where we have identified those translocations, and we know we realize the significance of these translocations. this is when we cannot classify AML as one with the recurrent translocations or one that is related with dysplasia or with uh, chemotherapy. So this 1517 translocation, why are we so interested in this? The 1517 translocation involves the fusion of the retinoic acid receptor gene. This RAR alpha gene is present on chromosome 17. This goes and fuses with the PML gene, PML in this case standing for promyelocytic leukemia. So, what is with this RAR gene? Now, you must all be familiar with the role of vitamin A in vision. What we also know is that the vitamin A has a very significant role in the differentiation of cells, including the hematopoietic cells. This translocation, which results in the fusion of the PML and the RAR gene, results in the formation of a fusion protein that does not allow the myelocytes to progress beyond the promyelocytes. What would this mean? This would mean that the myelocytes are all stuck at the promyelocytes stage. They accumulate in the bone marrow as promyelocytes, and now there are no mature neutrophils forming. So, how has this information been very useful to us? Knowing that this is what is causing the accumulation of the promyelocytes, the use of vitamin A, in fact, an analog, the all-trans retinoic acid, has proven effective in overcoming this maturation arrest or the differentiation block. So what this will do is these neoplastic promyelocytes will mature to neutrophils, and because we know that mature neutrophils don't live for long, they would all die. Something we might call death by differentiation. But in this case, we would be able to get rid of those neoplastic cells that are accumulating in the marrow, leading to the leukemia. Later on, the addition of arsenic trioxide has been shown to be even more effective than just giving vitamin A. It is not surprising that in AMLs where there are no translocations of this retinoic acid receptor, they obviously do not respond to the vitamin A treatment. Besides these mutations, there is also the IDH1 and the IDH2 mutations. What do these mutations do? They basically create the oncometabolite 2-hydroxyglutarate. 2-hydroxyglutarate inhibits some of the dioxygenases, and inhibition of these dioxygenases leads to the malignancies. And again, how is this information useful? Inhibitors of these mutated IDH1 and IDH2 have been shown to be effective in treating those acute leukemias where there are IDH mutations. Right, so this is another acute leukemia. Again, we can see the background RBCs much smaller, and we see a lot of these large cells. But unlike the acute myeloid leukemia we just saw a little while ago, these cells, very large cells, but also very, very large nuclei. The nucleocytoplasmic ratio is extremely high. Very little, if any, scant uh, we could say very little tiny amount of very pale looking bluish cytoplasm, and of course no granules whatsoever. So this also the flow cytometry. Most of the cells display positivity for CD22, a marker of B cells, TDT, which is a marker of primitive cells, and then of course 19, which is again a marker for B cells, CD10, a very specific marker for or the so-called CALA antigen, the common ALL antigen, a marker for B cell acute lymphoblastic leukemia. So just like the acute myeloid leukemias, the acute lymphoblastic leukemias also have some mutations that can be favorable, some mutations that are not favorable. So, what are the common mutations in acute lymphoblastic leukemia? One of the good mutations, let me start with this one, is hyperdiploidy. More than the usual complement of chromosomes in a cell. That's a good prognosis. 1221 translocation involving the ETV6 and the RUNX1, again favorable prognosis. Interestingly, the ABL-BCR translocation, which we will revisit a little while later with the chronic myeloid leukemia as well, it's not a good prognosis. And that is more often seen with the pre-B cell. Pre-T cell, we see translocations that involve the T cell receptors, the transcription factor genes like BTAL1. So, as I said, the T1221 and hyperdiploidy, these are good prognosis. And many children, in fact, most children can have reasonable expectation of a cure, which is uh, which is a remarkable thing with malignancies. But some of the other factors that lead to less favorable outcomes include male gender children less than 2 or more than 10 years. And it's also very interesting that uh, the mutations that we see in children less than 2 years that confer a bad prognosis are different from the ones that cause a bad prognosis in children over the age of 10 years. The BCR-ABL fusion gene, or what we know better as the Philadelphia chromosome, is often seen in children above the age of 10 years and associated with a not so good prognosis. In children less than 2 years, it's mostly rearrangements of the MLL gene. Right, so just to remind you, acute lymphoblastic leukemia, acute myeloid leukemia. In both cases, we see translocations, we see multiple mutations. Some of these have good prognosis, some of these have bad prognosis. We specifically looked at the case of acute promyelocytic leukemia, where the vitamin A, the all-trans retinoic acid, ATRA, has been shown to be very effective in treating acute promyelocytic leukemia, because we now understand that the mutation, the translocation, results in a differentiation block which can be overcome by providing the retinoic acid. Among the chronic myeloproliferative disorders, there is polycythemia vera, chronic myeloid leukemia, myelofibrosis, and essential thrombocytopenia. So, what do we have here? We have a 33-year-old man who presents with a non-specific symptom, fever for 2 weeks. The labs are interesting. His hemoglobin seems not so, just slightly on the lower side. Platelets within the normal range, slightly on the higher side of normal, we could say. But this is interesting. The WBC count is high, and most of these seem to be mature neutrophils. There are some earlier forms, the band forms, metamyelocyte, there's also myelocyte, and interestingly more basophils than we see. Again, that's one of the basophils, and we see neutrophils that look almost like mature neutrophils. In fact, it would be very difficult to say otherwise just by looking at this slide. And precursor forms, band forms, metamyelocytes, and even myelocyte. This, by now, you would have realized is a chronic myeloid leukemia. You see an increased number of the myeloid series, but there is maturation all the way. The translocation that is associated with chronic myeloid leukemia is called the Philadelphia chromosome, the ABL gene, named after Abelson. Sorry, my writing is a bit slow with the pointer. Right, so we have the Abelson gene on chromosome 9. This goes and fuses with the BCR gene on 22, the breakpoint cluster region. This fusion results in the formation of a new tyrosine kinase. Tyrosine kinase, as we are aware, will cause more proliferation of the cells. In this particular case, the myeloid series. It seems to remove the growth dependence of these myeloid cells on the growth factors. So it results in a lot of proliferation. Significantly, though, it does not cause a maturation arrest. One of the major differences between acute and chronic myeloid leukemias, if you will recall, is that there is a maturation arrest in acute leukemia. Cells do not seem to go beyond a certain stage and they accumulate. Whereas in chronic leukemias, both myeloid and lymphoid, they seem to differentiate all the way, except that they are abnormal neoplastic cells. But there is no block in differentiation. Those are two major things that we see. This new fusion protein that will cause proliferation of cells, and even though these are completely abnormal cells, they seem to go through the entire process of differentiation, resulting in a very large number of seemingly normal looking segmented neutrophils. And it's not just an increase in the neutrophils, there's also increase in the platelets, basophils, eosinophils. So essentially everything other than the lymphoid series, there is an increase. We see 4 different images. The first 2 images focus on the erythroid series. This is the sort of erythroid cell that we might think of as relatively normal around nucleus, but this kind of nuclei, these are definitely abnormal, so they indicate some sort of abnormality within the erythroid maturation. This is a Pearl's Prussian blue stain, the blue dots forming a ring around the nucleus, so-called ring sideroblast. This slide C, something wrong with the neutrophils. They seem to be bilobed. The Pelger-Hewitt-like anomaly. And then the last image here, a megakaryocyte, very, very large giant cell, having 3 separated nuclei, completely abnormal. But together, these images tell us that there is myelodysplasia. So while this may have not progressed to an acute leukemia as such, there is definitely a likelihood of progression to acute leukemia if more than 20% blasts accumulate in the bone marrow. Recently, there have been some updates on the myelodysplastic syndromes. It seems that the preferred terminology would now be myelodysplastic neoplasms, and a lot of emphasis on the genetic criteria for defining a disease rather than to just use morphology or risk-based a risk-based approach. In fact, there was even a suggestion in a recent article that perhaps in the future we might be seeing more of uh, an attempt to classify diseases based on the genetic abnormality rather than the morphology or the organ of involvement. Of course, this might happen in the future, but it does make sense. And why does it make sense? Because the greatest benefit of understanding the genetic abnormality is to be able to devise more effective strategies for treating or managing the disease. And if there are different uh, cancers that respond to the same treatment because they have the same genetic abnormality, it does make sense. But of course, we'll have to wait and see what happens in the future. Um, for now, these are the myelodysplastic syndromes or neoplasms that have defining genetic abnormalities. As you can see, some of the more common ones, the 5q, the long arm of 5 deletion, the SF3B1 mutation, and the TP53 inactivation. All of them have significance. This is most often associated with the ring sideroblast, and therefore treatment with lenalidomide. TP53, very poor outcomes in those myelodysplastic syndrome. There have been a lot of mutations that are associated with myelodysplastic syndrome. These include epigenetic factor mutations. And what stands out is that the same epigenetic factors that are mutated in AML are, we observe the same mutations in these myelodysplastic syndrome. Explains why in many cases the MDS progresses to an acute myeloid leukemia. Besides these, there are the RNA splicing factors, mutations in those, mutations in transcription factors. Also, as we just saw in the previous slide, the P53 mutations, loss of function, inactivating mutations of P53 are associated with particularly poor clinical outcomes. And we see that even the primary MDS and the therapy-related MDS, they seem to have similar recurrent chromosomal abnormalities, particularly the monosomies or the deletion. So, moving on, another set of these hematopoietic tumors which are associated with specific mutations, the JAK-STAT pathway. In this particular case, what we see is the outline of a cell, that's the membrane, and you notice this growth factor EPO, erythropoietin, comes and attaches to the receptor. Once the receptor is dimerized and the signal is transmitted all the way to the nucleus, transcription happens, cell proliferates, right? But then there is a very specific mutation, V617F. What does this mutation do? This mutation allows the cell to divide without being stimulated by EPO. So now the cell is no longer reliant on the presence of EPO. It can keep dividing. It can keep making more and more cells, which is purposeless. There was no need for those cells to divide. But they will start dividing and eventually result in a completely purposeless mass of cells, the basic definition of a neoplasia. So, who is positive for this mutation, this JAK2V617F, almost all those with polycythemia vera. But it's not just polycythemia vera. We see this mutation with myelofibrosis also, essential thrombocytosis also. Although in not as many cases, it's not very clear why with there is a much closer association with polycythemia vera than with myelofibrosis. So this is another mutation that is of great interest to us. So how does this help us? Again, treatment. JAK inhibitors can be very useful in cases of polycythemia vera. Right, let's move on to the lymphomas now. So what have we seen so far? We have seen examples of mutations that happen with acute leukemias. Some are favorable, some are not so favorable. And then we have seen mutations associated with the chronic myeloid leukemias, with myelodysplastic syndromes. The lymphomas, again, a very large group of malignancies that arise from the lymphoid cells but outside of the bone marrow. A simple way of approaching lymphomas is to think of them as Hodgkin and non-Hodgkin, and so everything that is not Hodgkin becomes a non-Hodgkin. Non-Hodgkin we further divide into B cells and T cells, and like we do with the acute and chronic leukemias, the more immature are called precursor B neoplasms. The ones that arise from these cells that follow later are called the peripheral neoplasms. This applies to both the B and the T. Now, if we look at the origin of the lymphoid neoplasms, here is an interesting thing. You see, a lot of lymphomas seem to arise from the germinal center, and these are mostly B neoplasms. Yes, it is true that there are many other lymphomas arising from other B cells and less commonly the T cells. So, why is it so? Why so many lymphomas arising from the germinal center B cells? One of the reasons seem to be that in the germinal centers there's a lot of class switching and somatic hypermutation going on. But this, because these processes are mistake prone, the risk of a transforming mutation is much higher. Also, many of these mutations happen during the B cell activation and they involve the DNA breaks. It doesn't seem to happen with the T lymphocytes as much. And perhaps what is really something we should focus on is that many of these translocations involve the immunoglobulin loci. Now, we saw earlier the complete list of the non-Hodgkin's lymphoma, but let us take an example of three non-Hodgkin lymphomas that are associated with very specific mutations. The follicular lymphoma 1418, mantle cell 1114, Burkitt lymphoma 814. I think you all must have realized 14 is common. So obviously there is something that is happening on the chromosome 14. So what is happening on chromosome 14? Because these three lymphomas, Burkitt is a very aggressive, rapidly growing tumor. Follicular lymphoma, one of the most indolent, slowly growing lymphomas that we know. Mantle cell somewhere in between. So why, if all these three translocations involve 14, Why is there such a difference in the behavior of these non-Hodgkin lymphomas? Let's just start with understanding what a follicular lymphoma is. If we take this image in the middle as a representative of a normal lymph node structure with the germinal centers and the follicles in the cortex, you notice in one there is a lot more follicles than before. And not only in the cortex, but they're also invading the medulla. This is follicular lymphoma. This, on the other hand, a lymphoma that has completely involved the lymph node, what we call as a diffuse pattern of involvement. But let's focus on the follicular. So what's happening in the follicular? Here you can see a lot of follicles that are basically involving the entire length and breadth of the lymph node, the cortex, the medulla, almost the entire lymph node. So what's the translocation here? 14, 18. What's on 14? BCL2. BCL2 we know is anti-apoptotic. What does that mean? It means that if there is an overexpression of BCL2, the cells will not die, because especially when cells get older, they are sent to death through the apoptotic pathway. But because BCL2 will prevent precisely this from happening, there will be an accumulation of these cells. So why is BCL2 being overexpressed? Because it has come from the chromosome 18. I'm sorry, the BCL2 was here. It has come to the chromosome 14. So what is there on chromosome 14? Chromosome 14 has the heavy immunoglobulin chain locus. What is special about this? This is always getting overexpressed, and not only does it get overexpressed, but it also causes the gene next to it to get overexpressed. In case of follicular lymphoma, it is the BCL2. So we get something like this. This is a higher magnification of the follicular lymphoma. If we think back to a normal or a not necessarily normal, but a reactive lymph node, then what stands out is there's something missing in these follicles. There are a lot of follicles, no doubt, but there's something missing. What is missing? These cells. What are these cells? These are macrophages. Macrophages, as we know, have dual purpose. They can kill, but they also serve another important purpose, and that is of cleaning the debris. So why do we see macrophages in reactive follicles? Because in reactive follicles there is a lot of cell turnover. Cells are being formed, but cells are also dying, and those lymphocytes are dying from apoptosis, and macrophages are required to clear the debris. So in the second image, no macrophages. Why? Because cells are not dying. If cells are not dying, why do we need macrophages? So that's abnormal, right? So all these follicles are abnormal. That itself tells us there is something wrong with these follicles. If we do an immunostain, and in this case we are looking for the BCL2, you notice that in B, the entire follicle is lit up with BCL2, very strong BCL2 staining. This explains why there is a proliferation, why there is a tumor, why there is a growth. It's not because of proliferation, it's simply because overexpression of BCL2 is causing or preventing these cells from dying, and therefore these cells accumulate and result in the lymphoma. I'm sure all of you would would have recognized this picture, a tumor on the face in a small child, in a young child, and then this very classical starry sky appearance. It's of course the Burkitt lymphoma. Interestingly, macrophages, and just a lot of macrophages. So why in a lymphoma that we just saw, no macrophages, in this lymphoma, a lot of macrophages? Another translocation, again involving 14, but in this case, not the BCL2, the c-MYC from chromosome 8. So now c-MYC, as we know, is a transcription factor. When this is overexpressed, it will lead to proliferation, excessive proliferation. It is not at all surprising that it is considered to be the fastest growing human tumor. So let's just go back a minute. When BCL2 is overexpressed, we get a lymphoma that grows very slowly because it's only growing due to the fact that cells are not dying. Whereas the Burkitt lymphoma, on the other hand, one of the most rapidly growing lymphomas, because a different gene comes next to the IGH gene on chromosome 14. The third lymphoma that I mentioned that also shares this 14 translocation involves zone 2. So if this is the follicular center, this is the mantle zone, and as you can see, there is a proliferation in this area, the so-called mantle cell lymphoma. What's the translocation here between 11 and 14? What does 11 have? Cyclin D1, again another gene that will promote proliferation. So where does this stand? This is somewhere in between the follicular lymphoma, which is a low-grade, slowly progressive, indolent lymphoma. In fact, one of the rare malignancies that has waxing and waning course. And on the other hand, Burkitt's lymphoma just growing so quickly. This is somewhere in between. So what we might call more of an intermediate sort of growth. Again, a very classical type of non-Hodgkin B-cell lymphoma. You see cells having cytoplasmic projections. We would like to refer as hairy projections, splenomegaly, middle-aged people. Very close differentiation for this tri-tap is myelofibrosis, but this is of course the hairy cell leukemia. Hairy cell leukemia, again, why do I mention this? Because it's just very interesting. This has a mutation called the V600E mutation, a mutation that we see associated with melanomas, bad prognosis. But the hairy cell leukemia has the exact same mutation, the V600E, good prognosis. Response to BRAF inhibitors, excellent response. One of the explanations for this is that it's probably not just the V600E mutation that is responsible for the poor outcomes in melanoma. There are a lot of other mutations that probably contribute to the dismal outcomes, especially when it has metastasized. Right, so to sum up, in all the Hodgkin and non-Hodgkin lymphomas, as we can see from this image, different mutations confer different features to the tumor. So with the Burkitt's, let's start with the Burkitt. MYC translocation, more cell proliferation. Mantle cell lymphoma, overexpression of cyclin D1, again more proliferation. Follicular lymphoma, on the other hand, BCL2 overexpression, less apoptosis. Germinal center B-cell lymphomas, because of class switching and somatic hypermutation happening more often in the germinal centers. Hodgkin lymphoma, the PD-1 ligand gene amplification, what does this do? This allows the tumor cells to escape destruction by the cytotoxic CD8 T lymphocytes. So in different scenarios, as you can see, with the different malignancies, different kinds of mutations, that lead to that allow the tumor to grow and establish itself. Right, so before I wind up my lecture, just a couple of questions I would like you to think over. So I mentioned the Philadelphia chromosome in chronic myeloid leukemia and in acute lymphoblastic leukemias. So there's one chronic leukemia, another one is acute leukemia. Interestingly, when there is the Philadelphia translocation, in CML it is associated with a generally good prognosis. But in ALL it is specifically associated with a bad prognosis. So begs the question, why? Why does the same translocation lead to different outcomes? That was one of the questions. Another one, three seemingly different tumors. One is a hematologic malignancy, the chronic myeloid leukemia. Another, gastrointestinal stromal tumor. And the third one, a dermatofibrosarcoma protuberans. It seems that they are completely unrelated, but there is something common to the management of these tumors, of course, based on our understanding of the genetic abnormalities. Right, so again, something to think over, yeah. And this is my list of references, useful resources that some of you may find useful in understanding more on the topic. And I would like to thank you all so much for your patience and listening to my lecture. I will say that I learned some new things while preparing this lecture, and I hope that you also learned something useful. So thank you all so much and have a good rest of the day.